Welcome to Biology 1440. I'm Professor Carafit, and this is the pre-lab video for Lab 1, Introduction to the Lab. Now, before we go into any of the lab material, I want to point out that there are some safety procedures we need to follow. So when you come to lab, make sure you have closed-toed shoes on. So no sandals or anything like that. If you can see your toes, you shouldn't come into lab, and that will be the same rule for almost all of your biology labs where you're meeting in the building. So keep that in mind. Closed-toe shoes. Also, we cannot have food or drink in the lab, so don't bring your water or your coffee into the room. Now, if you have a bottle zipped in your backpack, that's one thing. Of course, you could take it out and go in the hallway to drink it, but we can't eat or drink in the classroom. There'll be more safety stuff talked about in lab, but that's some of the basic lab safety rules. Now, another thing I want to point out is that you can typically miss up to 2 labs a semester. Once you've missed 3, you may be dropped from the course because that's over 30% of the labs typically in a semester, and you can't miss that many labs and stay in the course. So keep that in mind. And if you are going to miss, it's a good idea to let your instructor know because you're still responsible for the assignments for that week. So make sure you get your lab partner's contact information, things like that. So if you need to get lab data, you can do that and get your assignment written. Now, all assignments are done on your own, so don't do them with your lab partners because if you cheat, you could be dropped from the course or receive a 0 on an assignment. So you want to make sure you can work with your partners in lab, but out of lab, you need to do assignments on your own. So this week's lab is going to focus on data and how we present data, because without the ability to share data to another person, science kind of becomes pointless. So we need to be able to do that. And one thing we need to do in order to share data is pick a system of measurement. How are we going to present measurements? And what we use in biology and chemistry, etc., is the metric system. So make sure you are familiar with that metric system if you are not already. So there are base units, such as a meter, a gram, or a liter, and then there are basically changes of powers of 10. So, for example, a deciliter, a centimeter. So this is going to be a tenth of a meter, a hundredth of a meter, a thousandth of a meter would be a millimeter, all the way down to micrometer, which is a unit that we use quite a bit in biology, a nanometer, which is a billionth of a meter. So micrometer, a millionth of a meter, nanometer, a billionth of a meter. And it can go the other way. A kilometer is 100 meters, right? All the way up to giga, right? So you can go in either direction, and every change here is a movement of the decimal to the left or to the right. And a lot of times, errors that students make involve converting in the wrong direction. So which way do you move that decimal point? And there's information here on which way that would go. So pay attention to that. And here's just further information on that. One thing I want to point out that not all students are familiar with is that the symbol for micro, like micrometer, is this mu, this U shape, this Greek letter. That's an important one to understand. You'd see that next to M for meter, and then you would know it's a micrometer. Okay, nanometer is an N. Okay, so these are the units we'll most use in biology, for example. So the base unit, sometimes you'll see a kilo used, like kilometer, for example. But when we're talking about cells and life, we're usually talking about millimeters, micrometers, sometimes nanometers. Now, in the course, we are going to be making a lot of figures. And a figure is a graph with a caption. So a graph along with the written caption, and we'll never have you do a graph without a caption. So it's always going to be a full figure. And when you make a graph, keep in mind we set them up a certain way. The x-axis always contains the independent variable. So in an experiment, the independent variable is the thing that you can manipulate. So the thing you change. The y-axis is the dependent variable. It's the thing that depends on the independent variable, and that's on the y-axis. So if you were seeing how changing the amount of calories you eat impacts your weight, you can change how many calories you eat. That would be the independent variable. Then how your weight changes would be the dependent variable on the y-axis. So we're going to do a few scenarios in lab this week. You should read over them ahead of time. We're going to measure our arms and our legs to see if there's a relationship between the two, and graph that out. I won't spoil that for now, but what we're going to do is we're going to work together on those in groups, and you're going to form lab groups when you come into lab. You're going to work with your lab partners to answer some questions, to work through the graphing exercises. And some of the things we need to look at when we are graphing is how variable the data is around the average. And we'll do that sometimes with standard error bars, which we'll talk about more in class. But I also want to point out that mean, median, and mode can be quite important as well. Mean is another word for average. So I'm sure you're familiar with that. Median is the middle number in the data set, which number falls in the middle, and mode often is, well, mode is which number appears the most often, essentially. And they could, in a very normal distribution of data, like let's say we have student heights, let's say students' heights are measured and most students are this tall, and mean, median, and mode could be the same. But in a group where students tend to be skewed, so most students are this tall, but some are way taller than that, so this data set is skewed, and there's not that many, and in this case there's not that many very short individuals, then the mode might be here, the mean and median might be out here. So that can shift depending on how the data is distributed. We typically, we may not report on mean, median, and mode that much in class. We will report on mean a lot, but median and mode we don't always do. But keep in mind, they're not always the same number. It depends on how data is distributed. And the way that we tend to measure how spread out data is in the lab, how far things are from the mean, is the use of standard error of the mean. And if you've taken statistics, you'll be familiar with that. But we're going to learn to graph using Excel this semester and putting error bars on our data. All right, so once again, here's mean, median, and mode. Mean is the average, median the middle value, and mode is the most common value. All right, now lastly, once we've made a figure, so we have a figure, we then need to write a caption for it. And so we have a graph. We're going to add a caption to it that makes it a figure. And so to do this properly, we're going to start off first of all by not putting a title on top of the graph, but instead everything goes below the graph. And we'll start with the word figure and a figure number, typically figure 1. And then you'll have a sentence that is basically a title, a descriptive title, but concise. And so you're going to contain information on what is in that graph. You're going to have a second sentence where you describe the trends in the data, so the major things that happen in the data using quantitative wording, like there was a 35% increase between day 1 and 2 and a 28% decrease between day 2 and day 3, or something like that. And you're also going to have information within that caption, like what the dots are on your graph, or the bars. Are they averages, or are they something else? So mean plus and minus SE, that would mean that you have error bars on the graph, and you'll learn more about that in class. So SE stands for standard error. So this would tell the reader, hey, this graph has averages and standard error bars. And you'll tell them what the sample size was used in that graph. So how many numbers did you average together to get each of those bars or those dots on your graph? And so we are going to work on this in class, but I'd like you to bear in mind that there is an example caption in the back of the manual in the appendix that you can look at all throughout the semester to get an idea of what those look like. So here's an example of error bars and what they look like. So when I say plus and minus SE, error bars will look like this. So the average is the dot, and the standard error are the lines up and below that dot. And basically, error bars give us some idea of how different the data sets were from one another, how spread out the data were around that mean. The more spread out they are, the larger the error bars will be. So in this example, these two averages are different, but their error bars, so by error, I don't mean that we made a mistake. It's more of how spread out the data were. And if we look at this, we can see that these bars overlap quite a bit, which would tell me that these two dots probably are not significantly different from each other. In this example, these two dots might also not be significant from one another, but here, these likely are significant. The error bars don't overlap, so this data point and that data point, these data sets are likely different. Now we can do further statistical tests to find out if they are different. So we are always careful with our language. We would say these data points may not be different, and these data points may be different, is how we'd word that, unless we do further tests, like an ANOVA analysis or a t-test. One other thing I want to point out before we go, students often ask how to know what kind of graph to make. Well, if you have data in Bio 1 where you have continuous data on both axes, meaning numbers on both axes that are continuous, like 0 to 100 or whatever, that would be a scatter plot type graph in Excel, or a line graph. Okay? But if you have a category on the x-axis, like male, female, or before and after, and numbers only on the x-axis, then you're going to probably be doing a column graph. Okay? So that's how you know the difference. And there are examples in the first lab showing you the difference between the two kinds of graphs. All right, so I think that's enough for now. There could be a quiz in the first lab over this information, basic information on lab, nothing too difficult. But so read over your manual before class, read over the safety rules, and be familiar with those, and you should be good to go for class today.